What is going on, guys? In this video we are going to harvest some plants from a temporary immersion bioreactor. These plants are Drosera capillaris, a North and South American carnivorous plants. They have been growing in this platform bioreactor for 7 months, and honestly I should have changed the media or harvested the plants after the 3rd month as they haven't multiplied since then. I started the experiment with a small clump of plants from traditional tissue culture on April 22nd, but on April 30 it was twice as big. On May 13 it doubled in size once again, and by May 22nd, just a month after we started, it was about 10 times bigger than the original clump of plants. Finally, by June 22nd it doubled in size for one last time. After that, I should have changed the media or harvested the plants. It has been sitting in this container for the last 5 months doing nothing. So now let's open the container and take a closer look at the plants. They have hundreds of flower stalks and probably have over 500 or maybe even more than 1000 plants. There are some dead leaves, but most plants look great. I could have harvested the majority of the plants and leave a couple to keep the cycle going. However, I think it's time to experiment with different plants as well. So here is the deal, the new lab is almost finished, and we do have 10 of these platform bioreactors. We want to show you how to set them up and use them. We're probably going to do a weekly series setting up one at a time. So if you have any suggestions as to which plants you want us to experiment with, please let us know. You can leave a comment or send us a message on Facebook, Instagram or LinkedIn. If you want to learn how to do tissue culture from home, subscribe to the channel. Our goal is to make more educational videos. And if you feel that you are ready, you can go to plantcelltechnology.com where you can find all the material you need to get started.
Next, plants as a bioreactor. How we can use plants as a bioreactor for the production of different valuable products such as antibiotics, vaccines, antigens, biopharmaceuticals, biofuels, biomaterials, different types of industrial important enzymes. In recent years, the use of plants as a bioreactor has emerged an exciting area of research and significant advances in the creation of new opportunities. The diverging forces behind the rapid growth of plants bioreactor include low production cost. We can use the bioreactor plants as a emerging technology, phytoremediation. We can use the phytoremediation technique. We can remove the different types of environmental pollution from the soil, groundwater level, and we also express this transgenic plant to the different types of minerals, vitamins, carbohydrate, biopolymers also. Plants can be used as in the below way: seed-based plant bioreactor, plant suspension culture, hairy roots system bioreactor, chloroplast bioreactor, Types of plant reactor. It is an individual topic below the plant. Plants can be used as a below is seed-based plant bioreactor, plant suspension cultures, hairy root system bioreactor, chloroplast bioreactor. Some examples of vaccines, therapeutic proteins expressed in plants: epitope source, DPT polypeptide of Clostridium, diphtheria, Bacillus, pertussis, Clostridium tetani, microorganisms. E6 and E7 of HPV, hepatitis polyomavirus, PA of Bacillus anthracis, BP2 of CPV. On kinds of virus, FMDV, fluid mosaic virus, NP of H1N1 influenza A virus. We can use the host plant tomato, tobacco, and Bigna uniguiculata, hepatitis B surface antigen in maize, cholera toxins subunit B in maize, human epidermal growth factor in benthamania. Plants have a distinct advantage for the following applications. Plants can be used as a seed chemical factories that require only water, minerals, sunlight, and carbon dioxide to produce thousands of chemical molecules with different structures. Transgenic plants are attractive and cost-effective alternative to microbial system for the production of biomolecules. Advances in biotechnology are enabling plants to be exploited as bioreactors for the production of proteins, carbohydrates, and lipids. Not only might tailor-made saccharides and fatty acids, non-plant origin synthesis in plants, but overproduction of plants metabolites should be also feasible. So we can overproduce different types of metabolites, primary and secondary metabolites, by the use of plants as a bioreactor. Next, advantages and disadvantages of plants-based bioreactor. Generally, the practice has been used to clone animals to make antibodies for the use of drugs, but there is always remote chance of unwanted allergic reactions due to antibodies of animal origin that apart. So when we can use plants as a bioreactor, we also found some disadvantages and also some advantages also. When we use different types of plant-based chemicals and antibiotics and also metabolites, sometimes allergic reaction or hypersensitivity reaction may occur. And making recombinant proteins of transgenic plants is relatively cheap as compared to the cost of running fermenter, though extraction and purification have to be efficient. For example, these proteins are expressed in the plant seeds, which is or looks similar, and recombinant proteins have to be cleaved enzymatically by other means. So we can use plant-based bioreactor for the cost-effective methods from the extraction or purification of recombinant protein from other sources such as microorganisms and different types of animal sources. Production of plant-based vaccine. How can you produce plant-based vaccine by the use of plants as a bioreactor? The target sequence of the selected antigens is integrated with the vector before using transferred into the expressed system. Antigen like insulin, rotavirus, enterotoxin, anthrax lethal factor, HIV antigen, foot and mouth disease virus, antigen, heat stable toxin have been produced in plants. Next, the transgene is not incorporated into the genome of the plant cell. Finally, two most commonly used methods that would achieve transient expression of the desired protein in plants are agrobacterium mediated transformation of genetically modified plant virus and particle bombardment or biolistics method. So use the read some key points for the production of plant-based vaccine.